Perfect. All right. Well, thank you, Mona. Really appreciate the opportunity to talk to you all about kind of what I love, studying stroke in children and trying to understand plasticity. And I want to thank John for his great talk and sort of introducing this idea with his comments about some perinatal stroke. So this was really interesting, just kind of when Mona asked me to do this. I've always approached stroke as trying to understand what I can take from adult stroke and apply to children. And she wanted me to try to flip that around and say, what can we take from pediatric stroke and apply that to adults? So hopefully I'm going to be able to talk to you about some of my research and what we've been doing and how that might relate to recovery after stroke in adults. All right, so I have no relevant financial disclosures. And just to give you a little bit more background, Mona mentioned that I'm pretty highly engaged in both adult and pediatric stroke. And when I first started my training, it was kind of in the heyday of neural stem cells, and my image of myself in the future was going to be curing neurodegenerative disease in adults. And during grad school, I got engaged in kind of one of the only neural stem cell labs at my institution, which happened to be a perinatal investigation lab. So they were studying cerebral palsy, basically. That kind of led to my falling in love with pediatric stroke. Then when I got to residency, I fell back in love with adult neurology as well. So stroke really gives me the best opportunity to kind of live in both worlds. And I think I'm a bit of a strange person in that regard, because there's not too many people that I know that enjoy being on both sides of that age spectrum. But I think it's a lot of fun, and I learn a lot from both sides. So today I'm going to very briefly kind of talk about the epidemiology of stroke in children, just so that you understand what we're talking about, and really focus on what we understand about outcomes after pediatric stroke. And then we're going to shift gears and talk a little bit more about my laboratory work and introduce the idea of epigenetics as a potential regulator of neuroplasticity following stroke. So when we talk about pediatric stroke, often in research terms and in clinical terms, we often divide it by age, and we discuss perinatal stroke, meaning right around the time of birth, essentially, and then everything else, which would be childhood stroke from 1 month of age up through adolescence. And then we also talk about types of stroke, including arterial ischemic, cerebral venous sinus thrombosis, and hemorrhagic stroke. The point being that children can suffer all the same types of strokes that adults can, and we need to be thinking in those sorts of terms. A lot of what we understand is about childhood arterial ischemic stroke, and much of what I'm going to talk about focuses on that. So it's important to recognize that strokes can happen at any age, and the American Heart Association acknowledges this. They've got lots of cool little ad campaigns to help out with that. And I always like to show this picture. Who has a higher risk of stroke, the little baby or the gentleman on the left? And you might be surprised, but really the highest risk time of life for stroke is around the time of birth. The reason that we don't see many babies walking around with strokes, and we see a lot of adults suffering strokes, is that risk period is really compressed to just a few days around the time of birth, as opposed to once we get into adult life, that risk period exists each day, every day, all day, for many, many years. But children are at risk for a number of reasons. Perinatal stroke occurs in about 40 per 100,000 term births. As I mentioned, it's one of the highest risk periods of life for stroke. It's the most common cause of hemiplegic cerebral palsy, and the majority of these are ischemic strokes. And to understand why it's such a high risk period, we have to think a little bit about how babies are getting blood. And a lot of thought is that perinatal stroke occurs as a clot forming at the level of the placenta, traveling across the umbilical cord, and then in babies, the cardiac circulation has a patent foramen valve that allows an easy passage of blood from the venous circulation up to the brain and presenting a risk for stroke. The other reason that it's so common is that the coagulation cascade is upregulated right around term birth, both in mothers and in children. So mothers are at higher risk of stroke as well as babies. Everything is geared towards stopping bleeding as a baby's being delivered, because that's obviously a high risk of mortality and morbidity, is hemorrhage after giving birth. So it's a perfect storm setup for stroke around the time of birth. Now, childhood stroke, on the other hand, is actually quite rare. So after the perinatal period, the risk of stroke drops down to about 2 to 3 per 100,000, although some studies suggest that might be underestimated, and that we might be seeing incidence as high as 13 per 100,000. This is about similar to the incidence of pediatric brain cancer, which a lot of people acknowledge and recognize. Probably many recognize more than they acknowledge stroke. So it's important to understand that stroke can occur in children. And the causes of stroke in children are very different, right? It's not atherosclerosis and diabetes. One is cardiac disease, so congenital heart disease in children, as well as acquired heart disease, things like viral cardiomyopathies we see, procedures that are associated with cardiac disease, some arrhythmias we see mostly as a consequence of congenital heart disease. Arteriopathies are probably the next, if not the biggest cause of stroke in children. These can be acute things like arterial dissection from trauma. They can also involve unique entities such as inflammatory arteriopathies, something called focal cerebral arteriopathy, as well as genetic and more chronic arteriopathies that many of us will see, including Moyamoya disease and variants thereof. Cancer and tumors are commonly associated with stroke in children. Many remain cryptogenic even after extensive investigations. Infections, acute infections such as bacterial meningitis, tuberculous meningitis, viral infections, including herpes infections and varicella infections, are common causes of stroke in children as well. Thrombophilia probably plays an important role in some cases, at least as a supporting factor in combination with other risk factors. Sickle cell is a common disease. Fortunately, in the United States, we've implemented a lot of screening and early transfusion policies that have reduced the risk of stroke in those children. And then any other systemic illness, which can raise the risk of stroke in children. So one of the reasons I like to understand outcomes in children is because our perception is that kids do really well after stroke. And I certainly feel that following a lot of kids, you know, they seem to lead good quality of life after stroke, even after fairly severe stroke. So I'm going to introduce kind of what we understand right now about outcomes of stroke in children. And we're really far behind where we are in adults. We don't have the detailed studies that we have in adults, but we're starting to get there. First of all, just recognizing the societal impact. If you look at age groups published as the 10 leading causes of death, you see cerebrovascular disease is a leading cause of death in all age groups, all the way down to 1-year-olds, taking out the neonatal period. But so across the lifespan, cerebrovascular disease is an important cause of mortality. Now, a lot of the studies and the data that I'm going to be talking about come from a group called the International Pediatric Stroke Study, which we've been really fortunate to be part of since my predecessor, Laurie Jordan, was here, and then I sort of picked it back up when I got out of fellowship. This is a group of investigators really on a voluntary basis who are contributing to an ongoing registry of pediatric stroke. In the map up top, you see one of the most recent diagrams of where all of the participating centers are located. And you can see really it is worldwide, although Canada and the US comprise the bulk of the centers that are members of the International Pediatric Stroke Study. So this was a study by Lauren Beslow at CHOP looking at mortality after stroke in children. And over a period from 2003 to 2014, really there's not much change. It ranges from about 1% to 3% over that time period. If you look at in-hospital mortality rates for adults, about 2% to 5%. That's really not all that different comparing children to adults. I think where we see it, probably a big difference is sort of post-hospital mortality, I think takes a larger toll in adults than it does in children. Once children get out of the acute phase, they tend to do pretty well. And where we really started acknowledging deficits after pediatric stroke was in the earlier 2000s. This was a study out of the IPSS by Neil Goldenberg, who's now one of our colleagues down at Ball Children's Hospital. But they did a study. They looked at that time, they had about 600 or so patients in the registry. They found a 3% risk of death, consistent with what I had just shown you. And then at the time of hospital discharge, they had 75% of their children having neurological deficits. Now, the limit of this study is they really didn't qualify it any more than that. They just had some sort of neurological deficit, and that's really all we knew. So obviously, we knew we needed to do better than that to try to understand the impact that stroke has on children. And this is just to introduce, so obviously everybody knows there are thousands of outcome measures that we can use in stroke. In adults, we use the modified Rankin scale, which is really a functional classification based on, return to activities that you previously participated on. And that's challenging in children because obviously children are expected to attain new activity levels throughout lifetime. So comparing to their prior activity level is challenging and perhaps not valid. So a while ago, some investigators came up with a unique outcome measure to measure pediatric stroke, and it's called the Pediatric Stroke Outcome Measure. Essentially, it's a quantified neurological exam And if you look at the two boxes up top, it's divided into 5 subdomains: right and left sensory motor function, expressive and comprehensive language, and then cognitive and behavioral impairment. Each of those subdomains has a score of 0 to 2, and it's laid out as you see here, 0 being normal, 0.5 would be a mild impairment with no functional deficit, and then 1 or 2 based on the severity of a functional deficit associated with that. And this is basically compared to normal age-matched peers by a pediatric neurology exam. So that's been in use for a long time, and one of the problems with it is there have been varying definitions of what a poor outcome means with the PSOM. Some people would use a PSOM of 1 as a poor outcome, so having some moderate functional deficit, but you could also have 2 mild impairments with no functional deficit giving you a poor outcome. So more recently, we developed what's called a severity classification scale, and that's shown at the bottom. And it's a little bit complicated, but essentially it says it's based on normal, mild, moderate, and severe. And the gist of it is moderate and severe really is significant functional deficits. So either moderate functional deficit in multiple subdomains or severe functional deficits. And so this is something that we're moving more towards using this in terms of our outcome studies. And along with some other investigators in the International Pediatric Stroke Study, we published this study a couple of years ago now. We wanted to look at what were the outcomes across ages at 2 years after stroke. We'll get a little bit into what's the window for recovery in pediatric stroke, but we wanted to sort of figure out when a final outcome could be assessed, and based on patient outcome availability and things like that, we chose 2 years. This is just looking at the raw PSOM score at that time. The black bars are the frequency of scores in childhood stroke. The white bars are the frequency of scores in perinatal stroke. And you can see with the raw PSOM score, we have this large left shift. So fairly low numbers across most of the patients, but spread out with higher frequency, obviously, in our childhood strokes. So when we did that classification, we looked at perinatal at discharge versus 2 years, and then childhood at discharge versus 2 years. Perinatal stroke outcomes look fantastic, right? More than 60% of patients have normal functional status. The bulk of the others have mild functional status, both at discharge and 2 years. The one caveat here is, remember, we're talking about perinatal stroke. So 2 years after birth, these kids are only 2 years old. So things like cognitive and behavioral outcomes and language are pretty challenging to assess at that early time period. Childhood stroke, a little bit less good outcome at the time of discharge. So about 50% would have moderate to severe functional deficits. Obviously, we expect recovery over those 2 years, and you can see up to now about 70% have normal or mild functional outcomes at that point, but still 25% are going to have moderate to severe functional deficits after stroke at a time point of 2 years. And then you factor in the the low rate of mortality in pediatric stroke. This is a lifetime of neurological disability that these children are facing. So we really do need to figure out new methods of rehabilitation and trying to improve these outcomes. We did break this down a little bit more. So looking at larger age groups rather than just perinatal versus childhood, we looked at these different age groups. And I think this is important because there's this perception that younger is better, right? The brain's more plastic, it's going to heal better. And this data suggests potentially otherwise. So you see this is the left panel is looking at the raw PSOM score. The middle panel is looking at that severity classification score. And then it's broken down into motor, language, and cognitive behavioral domains there. And what you see is this U-shaped curve where it seems like some of the youngest children outside the neonatal period have worse outcomes than all other age groups. And physiologically, this makes some sense if you think about it, because that first year of life, first 2 years of life are important for a number of reasons in terms of critical periods of development. We have language coming online, we have motor control coming online during that time. And if you have an injury interrupting that period, you can understand why deficits may be more severe. So this is just kind of a simplification of how I like to think about functional outcomes. So when you have a stroke, there's 2 ways to get to your final outcome. One is recovery of impairment. The other is compensation adaptation. John really nicely outlined these. When I think about kind of early recovery, and if we think more about neuroplasticity later on, we're really trying to understand recovery of impairment. How can we get back to more normal ways of doing things versus just compensating and adapting? But really, in the early phase, we want to focus on recovery of impairment. We can always compensate and adapt, but it may be that there's windows of opportunity where we might actually be able to influence true recovery of impairment. This is a study many people are probably familiar with, the Copenhagen stroke study. This is really kind of where one of the ideas in humans that we have a window for recovery. So they looked at Fugl-Myers scores over, you can see 18 weeks after stroke. And patients basically recovered to a plateau around 3 months after their stroke, with a bulk of that recovery coming in the first month or so. So this comes with the idea where we have a window of several months where true recovery of impairment is possible. We try to get at this a little bit in our children and try to look at, does age influence recovery? So this is a complicated graph from that same study I've been talking about. Each dot here represents an individual child. Panel A is children who were normal at discharge. Panel B, representing the dashed line, is moderate or mild deficits at discharge. C is moderate deficits at discharge, and D is severe deficits at discharge. And then we plotted them according to their outcome at 2 years. Couple of things that popped out here. One is a phenomenon that we all understand is emerging deficits. So if you look at kids who were normal at discharge, this is the bulk of the perinatal strokes and the early strokes. They got worse over time. And we expect this to happen because motor deficits and language impairment and things like that really can't be picked up until later on. So when you have a perinatal stroke, these children really look quite normal at birth, and it's not until about 4 to 6 months of age that they start to manifest their hemiparesis. And then obviously we can't really be assessing many language or cognitive deficits until later on. So this is an expected finding. But if you look at the rest of the age groups, there's really no pattern that can be discerned over time. So age really doesn't seem to influence the degree to which these children recovered over time. This is another study from Ann Gordon's group looking at rehab. And this is, again, not as good as the data we have in adults, but they were looking at just the presence of any fine or gross motor deficit in children. They looked at groups of neonates, preschool age, or school age over time. And what you see is over the first 6 months, we get a reduction in the number who are showing gross or fine motor deficits. And then in most cases, we don't really see much change between 6 and 12 months. Again, not great, not really granular data, but it does suggest that maybe there is this plateau phenomenon and the kids aren't recovering very much after a certain period of time. And this is really where my interest focuses, is what characterizes that window of opportunity that we have to target rehabilitation. This is from an old study that really kind of characterizes what makes that up. This critical period after stroke is characterized by an upregulation of a number of growth factors that then changes to a more inhibitory environment over time. And if we think about it, the immature brain really represents an opportune time to study this because it's a growth environment to begin with. We can really start to take advantage of some of the plasticity that exists. So what is neuroplasticity? It's the ability of the brain to adapt. And we see this through neurodevelopment. We see this through learning as adults. We see it in responses to injury. And it involves a lot of mechanisms: axon growth, synaptic remodeling or reorganization, although after John's talk, I'll have to sort of reterm that as synaptic strengthening, I would say, and neurogenesis. And the degree to which all these mechanisms play a role still kind of remains unknown, but we'll move on. And one of the big focuses that I had throughout my career is neurogenesis. Again, I talked about how I was interested in stem cells, and we have a lot of data from animal models that neurogenesis correlates with outcome. This is just a review of a number of studies looking at various outcomes in animal models of stroke. And all of these showed a correlation one way or the other with neurogenesis. If there's increased neurogenesis, they had better outcomes. If there was decreased neurogenesis, they had worse outcomes. But the mechanism by which neurogenesis influences outcome remains a mystery. Many of these cells that are generated after stroke don't persist. They don't integrate into circuitry, although some can. So it's unclear whether they're providing a trophic support or what's going on. And when I first started looking at this, we were using a perinatal model of hypoxic ischemic brain injury. It's the classic Rice-Benucci model, which involves ligation of the right common carotid artery. We then subject those animals to a period of hypoxia, And this results in a hemispheric infarct, essentially, with a relatively normal contralateral hemisphere. So this isn't quite a focal ischemic stroke, but it's similar in that it is a unilateral infarct that's produced. And what we did is we isolated the subventricular zone, which is one of the neurogenic niches in the rodent brain. And we looked at the ability to form these clusters of cells. So if you dissociate the cells into individual cells, those neural stem cells will actually grow into colonies that look like this, called neurospheres. And as we looked at this over time, after 3 days following the stroke, in the injured animals, we got a big boost in the number of cells that were forming these colonies. So we seem to have an activation of the neural stem cell population. And what's more, when you allow these colonies that have been generated by a single cell, when you allow them to differentiate, they have the potential to differentiate into all three, broad categories of cells in the nervous system: astrocytes, oligodendrocytes, and neurons. A and B are sort of sham and contralateral hemisphere. And what we see is under baseline conditions or control conditions, the vast majority of these colonies are not forming all three cell types. Most of them are forming astrocytes only. A lot of them are forming astrocytes or neurons, but you don't have very many true multipotential cells. Following stroke, however, we get a huge burst in the number of colonies or expansion in the number of colonies that are actually generating all three cell types. So not only is the neuroprogenitor population being expanded in the wake of stroke, but it's also changing the behavior of these cells. They're able to generate different types of cells at a higher rate than previously. Further studies that we did identified Notch signaling as an important component of this. So we recognize that Notch is upregulated after stroke. And then we used a chemical called DAPT, which is able to inhibit cleavage of the Notch protein and essentially inhibits activation of the Notch pathway. And when we looked at the increase in neurogenesis, obviously when we administered DAPT here, we got a reduction in the number of neural stem cells that we were getting after stroke. Similarly, if we looked at the differentiation potential of those cells when we administer vehicle, again, we get this huge population that's multipotential. When we block Notch signaling, we're back to more of the astrocyte-only population. So Notch signaling seemed to be important in that study. So this leads into the idea that gene expression is important for regulating what's going on in the brain after stroke. And this, as I got into residency, I found this study by Hongjun Song, which led me to his lab as I was starting to train. And what they really started looking at was the idea of DNA methylation, and they were interested in more hippocampal neurogenesis and electrical stimulation. And this is a complicated graph, but it's an interesting study. So they have this protein GAD45 that's induced after electrical stimulation. You see 1 hour after ECT, they're getting this huge burst in GAD45 expression. When they looked at DNA methylation of some specific genes, BDNF and FGF, if you knock out GAD45, you get a reduction in the expression of these genes following ECT. So it seems like GAD45 is important for the expression of specific genes, but not all genes. They didn't see the same thing with FGF and OCT4. At the same time, when they knock out GAD45, they saw a reduction in neurogenesis following ECT. So here in wild type animals, you apply ECT, you get a huge burst in neurogenesis. We can see that response is attenuated in the knockout animals. So this suggests that GAD45 is important for activity-induced neurogenesis. And it really led me to question, okay, well, if it's important for activity-induced neurogenesis, maybe it's important for injury-induced neurogenesis as well. And that gets us into epigenetics. Epigenetics is really the key to unlocking our genome, right? Everybody has the same genes at all times throughout their life, but they're expressed in different ways throughout development, throughout aging. And to some degree, it can still be a mystery. I always like that comic there. But the big categories that people typically think about are DNA methylation, histone modifications, and then things like post-transcriptional regulation in terms of regulating the genome. DNA methylation is what I've really latched onto, and I think is an opportune mechanism to study plasticity. Just briefly, either in review or introduction, this is kind of the generic landscape of a typical mammalian gene. So we have, at the site of gene promoters, we have these CPG islands that are highly enriched in cytosine guanine dinucleotides. And this is where the bulk of methylation occurs on cytosine residues. If all these cytosine residues are methylated, then that blocks transcription. The transcriptional machinery can't access the promoter and the gene can't be transcribed. So methylation equals gene silencing. Unmethylated CPGs obviously allow access to that promoter region, and that allows gene expression. And for a long time, it was thought that DNA methylation was, a very stable silencer of gene expression. Once DNA was methylated, that was its baseline state. And now we understand that it's actually a much more dynamic process, and it can actually be reversed or demethylated actively. I'm not going to terrorize a lot of people with biochemistry, but this is just basically the mechanism by which cytosine gets methylated and demethylated. So we have DNA methylation enzymes that take cytosine, add methylation to it. And then through a series of oxidation steps, mostly regulated by a family of enzymes called TET enzymes, the 5-methylcytosine is oxidized to 5-hydroxymethylcytosine, and then a number of other intermediates before it's recognized as an abnormal base, excised, and replaced with unmethylated cytosine. So this is the biochemical way that DNA becomes demethylated. We know that GAD45 physically binds to TET1 and can facilitate this process. And so that's the importance of GAD45 in this process. It seems to facilitate TET1 demethylating DNA. So we started by looking in the same Rice-Benucci perinatal hypoxic ischemic brain injury model. We just looked at expression of GAD45 following injury. And we can see a large upregulation of GAD45B within 4 hours after the injury. This stays upregulated at 24 hours, while some of the other isoforms are downregulated at that time. This is an immunohistochemical picture looking at the subventricular zone. So this is the lateral ventricle of the mouse. This highly cell-dense area right here is the subventricular zone. That's the putative neurogenic niche in the rodent brain. The blue is DAPI, so that's just staining cell nuclei. The pink, which is the important picture here, is 5-hydroxymethylcytosine, so that intermediate step between methylcytosine and unmethylated cytosine. And what we can see here is these red cells that have their cell body adjacent to the ventricle and are extending processes up through the brain. These are GFAP positive cells in the subventricular zone, which many have identified as multipotential progenitors. And in panel A, this is a sham animal. If we look here, there's some expression of 5-hydroxymethylcytosine, but in the injured animal, it's much more intensely stained, and it really seems to be localized around this layer of cells that are neural stem cells. So we tried to look at this another way, in which we took the tissue out and we just measured the levels of 5-hydroxymethylcytosine. And we also looked at it, trying to quantify that immunohistochemically. And this graph essentially shows that there's really no difference between hypoxic ischemic animals, whether it be GFAP positive cells or GFAP negative cells, compared to sham. So if we look at a global level, we're really not seeing any difference in 5-methylcytosine or 5-hydroxymethylcytosine. But we know that the response to at least activity didn't seem to be global. It was more gene specific. And so we started looking at some specific genes, starting with BDNF, and this was just a technique called methylation specific PCR. Looking at ipsilateral, so the injured animals, the contralateral hemisphere, and then sham animals. At 4 hours, we have no difference in methylation of the BDNF gene, but by 24 hours, we see a reduction in methylation. So the BDNF gene seems to be demethylated in the ischemic brain compared to controls by 24 hours. And this kind of presented a dilemma. Okay, so we know there's lots of genes that are upregulated and downregulated. Are we going to sort of pick and choose individual genes and hope that one of those seems to be the key, or do we want to look more broadly? And so we decided to take a more unbiased approach and look at whole genome methylation sequencing. This is just a diagram showing the methods by which we can do this. You can essentially protect unmethylated cytosines through biochemical methods, either with this chemical bisulfite or by an enzymatic method. And then you can actually change the hydroxymethylcytosine or the methylated cytosine to a different base. And then so when you use next generation sequencing, you can detect changes in that sequence based on the methylation status of the gene. And we did this, and we chose a differential methylation ratio of 20%. And what we found was this, is that following injury, you can see there's almost a bell-shaped curve. So we have many genes being showing increased methylation following injury, and then many genes showing decreased methylation during injury. So this partially explains why, if we just look at a global level, we're not seeing much change in the level of 5-hydroxymethylcytosine or methylcytosine, because you have processes going in both ways. Sometimes some genes are being methylated, some genes are being demethylated. And what was fascinating is when we looked at the genes that were being demethylated, we ran some pathway analysis of the most demethylated gene categories. So we even increased our rigor to a differential methylation of 30% for this aspect. And these are the pathways that seem to be involved. So we had 173 genes that were 30% demethylated compared to the sham control. This identified 36 pathways. Alzheimer's disease or presenilin, which is the presenilin is responsible for cleavage of Notch pathway and activation of the Notch pathway. Angiogenesis, cadherin signaling pathway, which is important for neural stem cell regulation, FGF signaling pathway, which is important, IGF interleukin signaling pathway, which again is important in neural stem cell regulation, Notch signaling, PDGF signaling. So a lot of growth factors, a lot of things involved in progenitor cells or presumed neuroplasticity seem to be demethylated in setting of injury. So this was really encouraging. And then so we took or simultaneously, really, we were looking at the next step. So we were using the GAD45B knockout model and essentially performing that same experiment, looking at neurogenesis following injury, the same experiment that they did following electrical stimulation. So in this model, we induced the hypoxic ischemic injury in P10 animals. We then allowed them to recover 7 days. And at 7 days, that's really kind of where we see the peak of neurogenesis after injury, both in immature animals and in stroke in adult animals. And so at that time, we administered this chemical ethinyl deoxyuridine, which essentially labels dividing cells. We sacrificed the animals an hour later, and then we looked under the microscope at which cells were dividing. So that's the pink cells here. Again, this is the lateral ventricle. This cell dense region is the subventricular zone, and then extending out to this dorsolateral corner where in rodents, we have the rostral migratory string, which is a region of lots of neural progenitors that are migrating to the olfactory bulb. In the wild type animals after stroke, we see a lot of cells dividing following injury. So all the pink cells that we see here. When we knock GAD45B out, you see a large reduction in the number of those cells that appear to be dividing, and that's quantified up here in the graph. So in wild type animals, we get this boost in neurogenesis. We still see some boost in neurogenesis in the sham animals, or in the GAD45B knockouts, but it's not as significant. Using ANOVA, this is the only significant result that we had was the sham HI condition. So it seems that similar to the case where GAD45B is playing a role in activity-induced neurogenesis, we're also seeing a similar role in injury-induced neurogenesis. And so this is kind of the working hypothesis that we have. GAD45B is induced by stroke or other brain injury. This sort of migrates to the promoter region of the genes and facilitates TET-mediated demethylation of the promoter regions. This in turn upregulates growth factors and leads to mechanisms of neuroplasticity. These, the data that I've shown here are related to neurogenesis, but I think further questions will need to be investigated, whether it involves other mechanisms of plasticity, and we're hoping to be able to do that in the future. So the question is, does this mechanism apply only to the immature brain, or is this playing a role in adult animals as well? And I think we do need to be careful. So I'm just going to show this one slide from Tom Carmichael's group a few years ago. Essentially, they took cortical neurons in animals with stroke, and this is a very complicated graph, so I'm only going to focus on one thing. And they compared adult stroke animals to non-stroke immature brain, essentially trying to show, is stroke recapitulating a developmental process? And this is just looking at differential regulation of gene expression. And if you look at stroke versus control, stroke plus this GDF10 molecule that they were using to study recovery, there's very little differential gene regulation. It's actually quite a lot, but if you compare it to the other conditions that we're going to be looking at, there's very little. If you look at the P4 versus all of their stroke population, there's a huge differential gene expression. So essentially showing that stroke in the adult animals is very different from the developmental environment that you're seeing in the P4 brain, at least in terms of gene expression. So we can't think of the adult brain as simply recapitulating what's going on in development. But this doesn't address the question of, what gene expression changes are induced by injury. That question remains unresolved, and we should be looking at that in adults compared to the immature brain to try to understand where those differences are and how we can harness the mechanisms of neuroplasticity that we see in the young brain. So in summary, while children are widely perceived to fare better after stroke, neurological impairments are certainly common and they can result in lifelong disability. The immature brain, in my opinion, represents an ideal substrate to study repair and regeneration because it's already a growth environment. A lot of these mechanisms of growth are already at play because the brain is developing and we have critical windows opening and closing already. Neurogenesis is one mechanism of plasticity, and it does increase following a period of following brain injury. It is associated with better behavioral outcomes, but nobody really knows why yet. But I think using it as a model of neuroplasticity remains important. And then if we look at demethylation, DNA demethylation occurs in growth-related genes following perinatal HI. And I think this could be a key to induce neuroplasticity following stroke. Interestingly, if we go back to one of the questions that came up, for example, about psychedelics, there is emerging data that psychedelics in adults actually influence DNA methylation as well. So, some more anecdotal suggestion that DNA methylation might be an important regulator of neuroplasticity following injury. With that, I'd like to wrap up my talk and hopefully I can answer some questions or engage in some discussion. Also, sorry, forgot I want to acknowledge everybody who was a part of this work. So Steve Zeiler was already mentioned. We share a laboratory and have kind of similar interests in terms of recovery after stroke. TJ and Vincent were two people, undergrads and lab techs, who really did a bulk of the work on the laboratory stuff that I showed. My research mentors, Hongjun Song and Francis Northington. Some folks at the GRCF who really helped me with a lot of the genomic data, because that was a venture into big data that I hadn't taken before. My colleagues in the International Pediatric Stroke Study, especially Gabrielle de Weber, who's really kind of founded the field of pediatric stroke, I would have to say. And then Ravian and Alex, who coordinate the International Pediatric Stroke Study, and my funding sources. A lot of this was done during my KO8 award. Thank you very much for listening.